Hello everyone, in this video we're going to be talking about something called active transport, and I'll outline this method of transporting molecules from one place to another, and then we'll talk in more detail about its role in the absorption of glucose. Because when we eat foods that are carbohydrate rich, then we break those carbohydrates into simple sugars like glucose. We absorb the glucose normally into the blood from the lumen of the small intestine, but then we rely on this active transport to get into the blood the rest of our glucose. So we're going to talk about that as our example. So let's think about active transport initially as a process. Now, there are three key things to remember when we think about active transport. It's a process that enables us to get, if we just draw very basic animal cell there, cell membrane nucleus, active transport allows us to get molecules into our cells or across from one substance to another against a concentration gradient. So I've said in another video that diffusion of molecules happens from regions where there's a lot of them to where there's little, so things move from high to low. The key thing about active transport is that you go against the gradient. So molecules move from an area of low concentration to high concentration. So active transport happens when you go against the concentration gradient. The second thing to notice or to note is that only specific molecules are transported in this manner. So it's for specific molecules. If you have, for example, when we look at glucose a bit later on in this video, you can see that it can travel through carrier protein channels by something called facilitated diffusion. Active transport is for very specific molecules in certain instances where you have that opposing concentration gradient. And the third and most important thing is that it uses energy. Now, it uses energy in the form of what's called ATP, or adenosine triphosphate, which I talk about in more detail in a separate video. So the key things about active transport, as opposed to basic diffusion, let's say, is that it uses energy. It's not a passive process, it's an active process. So you have to use energy to go against the concentration gradient. You go from, and I'll just add this at the top, you go from low concentration to high, whereas most things tend to go from high to low. So let's look at the process of active transport in a bit more detail, because with this picture of the animal cell and the blue arrow, it seems very generic. So what we're going to do is go into a bit more depth about actually what happens at a membrane when we get active transport. So for this, we're going to need a bit of a diagram here. So I'm just going to draw, it's a very rough sketch. You might remember from a previous video that I've done on cell membrane structure, we have a phospholipid bilayer with hydrophobic tails, and that's what I'm just drawing here. So all of these circles, I'll even colour them in, I'll just put a bit of a yellow colour on them to match my previous video. So what I'm colouring in here are the hydrophilic water-loving heads of these phospholipids, and that you can see the tails are pointing inwards. This makes up a bilayer of our membrane, and then right in the middle, and I'll just shade it in red, we have what are called transporter proteins. These are, as you can see, because they're spanning across the whole of this membrane that I've drawn, these are intrinsic proteins. So let's just orientate ourselves for a moment. If we imagine here is outside of the cell, And down here we have inside of the cell. The first thing that would happen is that these specific molecules would bind to receptors on the protein channel in the cell surface membrane. So specific receptors on these intrinsic proteins. If we imagine here's one molecule here, and it's going to move, and come and bind with receptors on the protein channel. That's the first thing that happens in active transport. So if we imagine this blue dot that I've drawn, let's say it's calcium, Ca2 plus iron. So active transport would be when we have more calcium in the cell. There must be more inside than there is outside if we're going to actively transport it in. So there's a low concentration outside. Inside the cell there's a greater amount of calcium, so we're going to actively transport it in. So the first thing that happens is the calcium comes to bind to receptors on that protein channel. We then have ATP, so this adenosine triphosphate, bind to the protein channel from inside the cell. So I'll just draw it as a block here. So we have this ATP molecule from within the cell bind to the protein channel. Then something interesting happens to that ATP. The ATP, just draw an arrow here to explain, that ATP breaks down into ADP and P with a little I. Now what that means is that adenosine triphosphate breaks down into adenosine diphosphate, releasing a spare phosphate, if you like, an inorganic phosphate. Now the ADP comes off, but the P, that inorganic phosphate, remains attached to the protein, and it causes the channel to change shape and open. So what that ultimately results in is, and I'll just quickly, rather than draw all of those phospholipids, I'll just draw the membrane as these two black lines here. But what we have is something like this. So we'll put in our protein channel there. And we have this inorganic phosphate remaining attached to the protein channel. Now when that channel opens up and changes shape, we can see that that molecule can now pass through the cell. So this molecule that was here, this calcium, is able to now pass through that channel and comes into the cell. So we've used ATP in an active process, and we've gone against the concentration gradient to actively transport this calcium ion, this molecule, into the cell. Now once that's done, the phosphate is released, and you can imagine that if you have a phosphate, so this green P that I've drawn, if that's been released and then you have a spare, if you have ATP just floating about on its own, you can see that that phosphate will go and rejoin with the ADP to reform our ATP. So we're not completely using our ATP there. That P, that inorganic phosphate, will rejoin with the ADP to reform ATP. The protein channel, when the P gets released, when that phosphate comes off channel, will close, and the whole thing can start again. So we'll have number 1 happening, where we have the calcium joining. Then I should have added number 2, which is where the ATP binds. ATP, then number 3, breaks down into ADP and the organic phosphate that remains attached. That's 4. And then finally number 5, the attached phosphate causes the channel to open and allows the molecule to pass against the concentration gradient to its desired location. Now that ultimately is how active transport works. What I'm going to do in the remainder of the video is talk about its role in the absorption of glucose in something called co-transport. So what we'll do is reduce the screen down again. Rather than put this on another slide, I think I'd rather have it all on the same thing so we could see what I've done previously. So we're going to imagine, think of our small intestine. Now this is only a very rough sketch, but we know we've got those finger-like projections. So we've got a network of capillaries and blood vessels. We've got finger-like projections in the small intestine called villi. Now I'm not drawing out the structure of a villi, but what I'm going to do is put in place a sort of a diagrammatic representation of three interfaces that we have. So I want you to imagine that we have sort of three sort of layers here. This top part represents the lumen of, and I'll just put SI, the small intestine. So this top part represents the lumen of the small intestine. What we have in the second layer, if you imagine it, this is an epithelial cell of a villi, or villus rather, single villus. And this third layer, if you like, is going to represent our blood. So we have the lumen of the small intestine, the epithelial cell on the surface of the villi, and our bloodstream. Now when you have glucose normally in the lumen, it will just diffuse into the blood, across through these layers into the blood, because there's lots of glucose in the small intestine compared to the bloodstream. So via diffusion, it will move with that gradient. But there comes a point when there's now more glucose in the blood than there is in the lumen. So we have to actively transport, or make use of active transport to get the remaining glucose in. And we get something called the co-transport of glucose, because we get glucose to come in at the same time as sodium. So it's called co-transport. We transport glucose with something else. So the first thing that happens in co-transport of glucose is that sodium, so we have sodium ions here, Na+, sodium ions get actively transported. And I'm just going to put, this is only a very rough sketch, but I'm going to just put some channels in place here. Sodium gets actively pumped out of the epithelial cell into the blood. So it gets actively pumped into the blood. Now there is, we could add this level of detail, but to balance that you can have potassium ions coming in to counter that effect. But I'm going to leave potassium just at the moment because I really want to focus on the sodium aspect. By initially, we'll call that number one, by having sodium actively being pumped from the cell into the blood, even though there's a high concentration in the blood, what that does is lower the concentration of sodium in the epithelial cell. So we have some glucose, we'll write this in red, we have, and I'll call it G for glucose, we have some glucose in our lumen of the small intestine. We also have some sodium, but we've pushed sodium out of the epithelial cell. So we've lowered the amount of sodium in that cell. So we have a higher concentration of sodium in our gut, in our lumen, than we do in the epithelial cell. So there's more sodium in here than there is in here, essentially. So sodium can come into that villi, the epithelial cell of the villi. And when it does, it comes in with the glucose. So when we actively transport sodium out of the cell into the blood, it creates a lower concentration in that epithelial cell, in that second region there. The sodium then combined with the glucose will move with the concentration gradient So both come through into the epithelial cell from the lumen. So by pushing sodium out, we've created a concentration gradient where it can move from high to low, and it comes in adjoined to glucose. So they both come through that protein channel via that co-transport protein. Once in the epithelial cell, those two can separate. So you have the glucose here, and you can have this sodium here, and you'll see now that we've replenished our sodium, we've restored the concentration of it in that epithelial cell. So we originally actively transported it out, but now we've taken it or replaced it and put it back, restored it to our normal concentration by bringing it in from the lumen. But what we've been able to do is pull glucose from the lumen of the small intestine into the epithelial cell. Once we've done that, the glucose via facilitated diffusion using another protein carrier, another type of protein carrier, is able to pass from the epithelial cell into the blood. So that's by I put FD, that's by something called facilitated diffusion. And with the number 1, it's AT, active transport, that is involved. So we actively transport sodium out. That's the first stage. Then sodium coupled with glucose will come in from the lumen into the epithelial cell, and then finally glucose, once detached, by facilitated diffusion will pass from the epithelial cell into the blood. And that is what we mean by the co-transport of glucose. We can see how active transport plays a key role in bringing that glucose from the lumen into the epithelial cell and ultimately into the blood. Okay, I hope all that helps.